Hello, my name is Lorena Castle. Today's date is April 22nd, 2023. My Module 4 assignment is going to be on the movement of macromolecules inside of a cell. The movement of macromolecules inside of a cell can be defined as the routes that lead inward from the cell surface to lysosomes. This process is called endocytosis. The three macromolecules that I'll be discussing will be the steroid hormones, the protein hormones, and the intracellular proteins. The topics of discussion will be where the macromolecules are created, the specific routes from the beginning to the end, and the ultimate fate of the molecule. I'll start out by talking about the flow chart for each of the macromolecules inside of the cell. The first hormone I'm going to talk about is the steroid hormone. The primary hormones are derived from lipids, which are fats, and the steroids. Steroid hormones are derived from lipid cholesterol and are considered hydrophobic. They are synthesized in a series of reactions from cholesterol and binds to intracellular receptors to change gene expression directly. Steroid hormones require carrier proteins to travel in blood. Some examples of steroid hormones are estrogen, progesterone, testosterone, and cortisol. The hormone crosses the cell membrane. It binds to the receptor protein. It becomes the transcription factor for mRNA in the nucleus. The mRNA leaves the nucleus. Then the mRNA is read by ribosomes. Protein is established, and the protein is secreted by the plasma membrane. The ultimate fate of steroid hormones are secreted by numerous glands in the human body. Here is the action of the steroid hormone. As you can see, where it enters the hormone, enters the cell, and as you can see where it binds to the receptor proteins, and then eventually is secreted by the plasma membrane. The next macromolecule that I'll be discussing will be the protein hormones. Some examples of protein hormones are insulin, thyroid, and growth hormones. They are synthesized in cells from amino acids. Now, the first messenger binds to the receptors in the plasma membrane of the target cell. Then it activates a cascade mediated by a second messenger inside the cell. The ATP activates the enzyme, and then the hormone affects cellular function. The hormone receptor interacts with different molecules to induce a variety of changes, such as an increase or decrease of nutrient sources, growth, and other metabolic functions. Here's an illustration of the protein hormone action in the cell. The first messenger, it binds to that receptor protein, and then that enzyme is activated. And then now that second messenger has an effect on the cellular function, such as glycogen breakdown. Now the third macromolecule that I'll be discussing is going to be the intracellular proteins. Some examples will be the DNA polymerase, the DNA helicase, and the RNA polymerase. They are synthesized on cytoplasmic free ribosomes. What happens is that the protein will enter the cell and then is transported by the Golgi apparatus and the endoplasmic reticulum. Protein is released into the cytoplasm and then is transported into the nucleus where it regulates gene expression. Here's an illustration of the intracellular protein action in the cell. The fate of these proteins are absorbed and incorporated into the body without undergoing any marked chemical change. This will conclude my presentation on Module 4 on the movement of macromolecules in the cell.